Hello and welcome to the AI Research Roundup. I'm your host, Alex. Today is May 3rd, 2025, and we're discussing a paper titled, Learning to Discover Regulatory Elements for Gene Expression Prediction. This ICLR conference paper, published just over 3 months ago on January 22nd, introduces Seq2 Express, a deep learning framework. Its main goal is predicting gene expression by combining DNA sequences and epigenomic signals, focusing on explicitly learning and extracting the key regulatory elements needed for accurate prediction. Alright, so this first figure lays out the conceptual groundwork. It illustrates the causal links the authors are modeling, starting with the raw DNA sequence and those epigenomic signals. These inputs influence different types of regulatory elements, like ones identified by measurement, or ones interacting with the gene. Ultimately, it's the actively interacting elements that drive the final gene expression, which is the target Y that Seq2 Express predicts. Okay, building on that conceptual model, this next figure details the actual Seq2 Express pipeline. We see the inputs, the DNA sequence X_seq and epigenomic signals X_sig feeding into a generator. This generator combines information from both, using a beta distribution to learn a mask M for the actively interacting regulatory elements. Then, the predictor takes these selected masked elements, X circled dot operator M, to predict the final target gene expression, Y. Moving to the results, this table quantifies how well that pipeline actually works. It shows the performance on gene expression prediction for 2 cell types, K562 and GM12878, comparing Seq2 Express against several other methods. Looking at the metrics like mean squared error and Pearson correlation, well, Seq2 Express consistently comes out on top, achieving the best scores across the board, which really validates its approach to identifying those key regulatory elements. Okay, so beyond comparing Seq2 Express to other deep learning models, this table then details a comparison against MAX3, which is a more traditional statistical method for identifying these regulatory regions. We see the same metrics, MSE, MAE, Pearson, but also mask ratio. Well, Seq2 Express Hard consistently gets better prediction scores than MAX3. And importantly, it does this using a much smaller mask ratio, suggesting it's more effectively pinpointing the truly critical regulatory elements. Following up on those performance comparisons, this table gives us a look under the hood at the model's setup. It lists the key hyperparameters, like layers, dimensions, learning rate, and those important beta parameters, alpha 3 and beta 3. The bold values show the final configuration used. For instance, 4 layers for both generator and predictor, which produce the strong results we saw earlier when comparing against MAX3 and other models. Alright, so drilling down into the specific K562 cell type results, this table provides a more comprehensive comparison against various baseline models. We see Seq2 Express, both the hard and soft versions, listed at the bottom. Well, looking across the MSE, MAE, and Pearson columns, Seq2 Express Soft consistently achieves the best scores, highlighted in bold, clearly outperforming models like Caduceus with signals, EP Informer, and the DNA-only methods, reinforcing its prediction accuracy. Okay, so complementing the K562 results we just looked at, this table presents the performance breakdown for the other cell type, GM12878. It's the same setup, comparing Seq2 Express against the baselines using MSE, MAE, and Pearson. And well, the story is consistent. Seq2 Express Soft again achieves the top scores across all metrics, shown in bold, reinforcing that its strong performance isn't just a one-off, it generalizes well. Alright, so to bring it all together, we saw Seq2 Express deliver top performance for both the K562 and GM12878 cell types. Really, the main story here is how it effectively integrates DNA sequence with epigenomic signals, learning to discover those truly influential regulatory regions. This results in better prediction accuracy than baselines, and even traditional statistical methods. That's our roundup for today. Thanks for joining me.